This is Sarah Wilkinson from Humber College and the University of Guelph Humber, and in this video series I'm going to be talking about lactic acid as it relates to exercise physiology. This is the first video in the series. In this video I'm going to cover lactic acid formation in the skeletal muscle, as well as the fates of pyruvate and NADH from glycolysis in order to set up the rest of the videos. This basic knowledge about the rate of energy expenditure as it relates to exercise intensity will allow us to look at the factors governing lactic acid formation. And just a note, it's more than just a lack of oxygen. There can be other factors even if there's sufficient oxygen present in the mitochondria. We'll finish off the series looking at lactate threshold and exercise performance. Please ensure you understand the basic ATP producing pathways, specifically glycolysis, conversion of pyruvate to acetyl-CoA, Krebs cycle, and electron transport chain. Understanding these will be essential for understanding lactic acid formation. So let's get started. As a quick review, recall that through a series of 10 reactions in glycolysis, glucose, a 6-carbon molecule, is converted to 2 pyruvate molecules, so 2 3-carbon molecules. And as you can see here, we have a 10-step reaction that's going to both consume and produce ATP and is going to form a couple molecules of NADH. To review, products of glycolysis include pyruvate and NADH. So in the cytosol, hydrogen from NADH can be donated to pyruvate, and the product of that reaction is called lactic acid. As NADH is oxidized, its reduced form becomes available, so NAD. The key here is that NAD is now available for glycolysis as an electron and hydrogen acceptor. So this reaction is often written in this way, in that pyruvate receives hydrogen from NAD and becomes lactic acid, so that NAD plus is reformed. The enzyme for this reaction is lactate dehydrogenase, and we'll talk in future video series about this enzyme. So lactate dehydrogenase is the enzyme that catalyzes the formation of lactic acid, and this occurs in the cytosol of the cell, so outside of the mitochondria. Just to further illustrate what's occurring here, we have pyruvate and NADH and our enzyme LDH. And just like any enzyme, the two substrates bind to it, a reaction occurs, and we have 2 products that can then leave, and the enzyme is unaffected by the reaction. So as a review, lactic acid is formed in the cytosol. It's formed by the reduction of pyruvate, and the enzyme is lactate dehydrogenase. Let's move on to the fates of pyruvate from glycolysis. It doesn't necessarily have to become lactic acid, so we'll talk about circumstances that lead to the formation of lactic acid and circumstances that lead to pyruvate going somewhere else. So, as a quick review, glycolysis forms pyruvate and NADH. And the key point to this is that in order for glycolysis to continue, there needs to be NAD plus present. I'll say that again because it's really important. In order for glycolysis to continue to produce ATP, NAD needs to be present. So we need NAD. Now there's a couple ways in which NAD can be reformed, and we'll get to that in later videos. But let's just look at what can happen to either pyruvate or NADH after it's formed. So ideally, pyruvate is going to enter our mitochondria. It's going to go through a reaction and become acetyl-CoA, and that's going to be able to enter the Krebs cycle, which will produce some more NADH. In addition, NADH is hopefully also going to be going into the mitochondria as well. And as you can recall from previous videos, electrons from NADH are donated at complex 1. I'm not going to review what happens in the electron transport chain, but the most important thing here is that NADH electrons go into the electron transport chain and we have a resulting ATP. So a couple things to note here is that pyruvate and NADH are formed in the cytosol and have to pass 2 membranes in order to make it into the matrix. In addition, some reactions need to happen in the mitochondria. So if any of these slow down or have barriers to them, we're going to have a backup of pyruvate and NADH in the cytosol, and therefore no NAD for glycolysis. And we'll come back to this point in future videos. As a quick recap, there's 2 possible fates for pyruvate and NADH producing glycolysis. The first possible fate is for them to enter the mitochondria. In this video, I haven't covered how pyruvate and NADH end up in the mitochondrial matrix. That really is an entire video in itself. And for simplicity's sake, we'll just say they have to enter the mitochondria. The second fate of pyruvate and NADH is to form lactic acid in the cytosol. In a future video, I'm going to cover what determines whether these 2 products enter the mitochondria or where they become lactic acid. Let's build on our take-home points. That lactic acid is formed in the cytosol. It's formed by the reduction of pyruvate, and its enzyme is lactate dehydrogenase. There's 2 very important points that come out of this video for this lactic acid series. The first one being NAD is needed for glycolysis to continue. If there's no NAD, there's no electron and hydrogen acceptors, and glucose cannot be oxidized fully. The second point is that pyruvate and NAD need to move from cytosol to mitochondria to be used in aerobic ATP production. If there's any backup along the way, then there may be a challenge. So remember these 2 very important points for this lactic acid series. Please continue on to video number 2 in this series about the rate of energy expenditure and exercise intensity.